Okay. Ladies and gentlemen, so we are going to start with the class, and I hope you can now all see me. Okay, so let me share my slides. Okay, so ladies and gentlemen, you are all welcome, and I hope you can all see my slides. Can you hear me and can you see my slides? Okay, so what about my slide? Can you see my slide too? Okay, so please tell them in the group that those of you tell them on the WhatsApp group that they should use this link, the recent one that I sent. Please do that. Please send it in the group. Okay, ladies and gentlemen, you are all welcome to our today's class. And of course, in today's class, we are going to look at the concept of chemistry of amino acid and proteins. That is what we are going to look at, and I hope that the class will be interesting and you will also enjoy it. So, actually, in this class, as I said, we are going to look at the concept is actually about the chemistry of proteins and amino acids. And of course, in the concept of this chemistry of amino acid and proteins, we are going to start with the sources, the classification, and the structure of the amino acid. So these are what we are going to look at. So okay, I think let me just give me 1 minute, 1 minute before we continue. I wanted to do something. 1 minute, yeah. Okay, so I think we can start with the class. So as I said, we are going to look at the sources, classification, and the structure of amino acid. So first, what are amino acids? Amino acids are the building blocks of proteins. They are the building blocks of proteins. So when you said building blocks, it means that a protein is like a building. It is like a building. And in that building, you have the blocks that are needed to make the building. So the blocks that are used in building or in constructing the house, which is the proteins, are the amino acids. So you need the amino acids to build a proteins. And these proteins, as you can see from the slides, proteins are naturally occurring polymers that are composed of amino acids joined together or joined to one another by an amide or a peptide bond. So therefore, now what we are seeing here in this case is that when these amino acids are used in building the proteins, number one, you need you need a blocks, and the blocks are the amino acid, and these blocks are joined by what you call um, a peptide bond. So a peptide bond is the bond that are used in building or in joining the amino acid together. It's like I can see that, okay. No, I think the problem is from you because from this side, the slide is very clear, and I hope that some of you that have a good network, you can see everything. Can you see it? Okay, so as I said, that proteins are the naturally occurring polymers that are composed of amino acid, and these amino acids are joined to one another by an amide or a peptide bond. So as we are saying, actually, when you are trying to have a protein, so you need a building blocks, and these building blocks are amino acids. And how these amino acids join themselves together is through a bond. So there must be a bond that will join this amino acid, and this bond is called a peptide bond. And now, what I want us to understand is that we have a lot of examples of the proteins. We have animal hair, we have muscles, we have egg whites, and we have hemoglobin, and they are all examples of proteins. Like, for example, we know that hemoglobin is a protein that is found in the red blood cell. And of course, we have egg whites. It's also a protein content of the egg. Of course, in the muscles, we have what you call actin and myosin. They are all examples of proteins that are found in the muscles. And of course, in the animal hair, we have keratin. So these are actually examples of the proteins. And one thing that we understand, or that one thing that we should understand, is that these proteins is what make us to do all the activities that we are doing. As I'm talking now is because of the proteins. My eyes, I use my eyes to see clearly is because of the proteins that are there. So in fact, the proteins is what determine the physiological functions of each and every cell of the body. Like for example, we know that the red blood cell is responsible for the transport of oxygen. So but now the question is, why is that the red blood cell is transporting oxygen? It's because there is a hemoglobin. So it's the hemoglobin that make red blood cells to do that job. And in addition, white blood cell. The major functions of white blood cells is actually try to actually provide immunity to the body, to protect the body against any infections. So but you ask yourself, why is it that red blood cell is transporting oxygen? And why is it that red blood cell is transporting oxygen and the white blood cell is responsible for the immunity of the body? It is because of the proteins that are there in the two blood cells are different. Like for example, in the white blood cells, there are immunoglobulins, there are antibodies, and those antibodies are proteins. And that what make white blood cell to protect our body from any foreign bodies. And of course, the brain. Why is that the brain is responsible for our cognitive function? It's because of the neurotransmitters that are found there. And the neurotransmitters that are found in the brain, they are either peptides, proteins, or amino acids. So one thing that I want us to understand is that proteins that are found in the body are responsible for each and every activities that are taking place in our body. In fact, in most of the disease conditions, is because of the problems of the proteins. So if one protein is produced in smaller amount or in higher amount, that one also lead to disease condition. In fact, even in many cases, if you are trying to diagnose a disease, you test a proteins because in most of the laboratory tests that we are doing, is it must be either qualitative or quantitative. So the quantitative is that okay, so in normal sense you expect that the proteins will be there. But maybe when detecting, like for example, but when detecting the protein, you will check whether the concentration, that is the amount of the proteins, is high or low. But in the qualitative test, it's just for you to detect the presence or absence of the biomarker. So even in terms of the biomarker, most of the biomarkers that we use in the disease diagnosis, they are proteins. So that is why understanding the concept of protein is very, very important. So proteins is important, and as I said from the beginning of the lecture, that the proteins are made up of amino acids. So in one proteins, you can have more than 100 amino acids. And how this amino acid joins to produce these proteins is through a bond, because there must be a bond that will link them together. There must be a bond that will be linking them together. So the force or the bond that linked one amino acid to another is called amide bond, or you can call it peptide bonds. And then in addition, we have what we call peptides. So is there any difference between proteins and peptides? You see, so the first thing that you need to understand is that look at the proteins. In the proteins, we said it's a polymers that compose of amino acids. But look at peptide, is the oligomers of amino acids. Oligomers. So when we said oligomers, actually the number of amino acids that are found in the peptide are less than the number of amino acids that are found in the proteins. So, and I think at this point, even from this point, before you go deep, you need to understand the differences between these three things. One, protein. Second, amino acids. And then third, peptides. So when you have a sequence of amino acids of between 2 to 50 amino acids, it is peptides. It is peptides. And when you have a sequence of amino acids of more than 50, so you have proteins. And of course, amino acids is the monomeric unit. So this is the three differences between the two. So whenever you ask what is the difference between amino acid, peptide, and proteins, I think you can clearly say that. Amino acid is the monomer. And then peptides is a chain of 2 to 50 amino acids. And a proteins is an amino or is a or is a sequence of protein, sorry, it's a sequence of amino acid with more than 50 amino acid. So I think from here you can actually understand the differences between the two. So then example of the peptides. Example of peptides, we have insulin. So insulin is an example of peptides because in insulin you have less than 50 amino acids. So that is why insulin is not considered as a protein. Rather, we call it peptides. But now the question is, you see that from the questions, you see that we have peptide hormone. So that is why I think you have this basic understanding, this general understanding that we have three types of hormones. We have protein hormones, and we have peptide hormones, and then we have steroid hormones. So now you know that insulin is a peptide hormone. And why is it that it is a peptide hormone? Is because the number of amino acids that are found in the insulin, they are less than 50. So I expect after this class is an assignment, I want you to go and check how many amino acids are found in insulin. And if you are able to find the number of amino acids that are found in insulin after this class, please write it on the comment section of this video. So that at least from there it will actually give you an idea of exactly the number of the amino acids that are found in the protein. So that as a nurse, wherever you are going to talk about the insulin, at least you have an idea that okay, insulin is a protein that is responsible for telling us the situation of a particular patient, whether it is diabetics or non-diabetics. Because we know that insulin is usually produced in a beta cells of the pancreas. So at this point, because we are doing chemistry of amino acid, go and know exactly how many number of amino acids are found on in the insulin. And then so amino acids are often from hydrolysis of protein. So if you want to get amino acid, how do we get them? Is through hydrolysis of the protein. So you break down the proteins using water molecule, and that is hydrolysis. This is breaking down hydro water. So hydrolysis means breaking down of the proteins with the addition of water. So you see in these slides, you should not forget, you shouldn't forget that we have proteins, we have peptides, and then we have amino acids. I saw someone writing 51 amino acid. Sit down and check very well. Okay. So then the next thing is, so you see that if you want to get the amino acid, then it's through hydrolysis. So proteins, peptide, and amino acid are essential to the structure, function, and reproduction of living matter. So you see here, we can see clearly the proteins, the peptide and amino acid are essential to the structure, function, and reproduction of living matter. So now, I said after the video, after the class. I'm not saying that you should be distracting. You should after the class, then you check and confirm. Okay. So proteins, peptide, and amino acid, they are very important in structure, function, and the reproduction of living matter. So, but now the question is, why only reproduction? So now the idea is not actually about reproduction. The proteins, peptide, and amino acid is responsible for every processes, or for every physiological processes that are taking place in the body. It's not only even physiological process, even the functional process, even the anatomical structure of living organisms. Because now the question is, if you look at the shape of the liver, the shape of the kidney, the shape of the pancreas are all different. So why are they different? It's because of the proteins. So the proteins that are found in these different organs is what make the different anatomical structure of each and every organ in a living organisms. So that is what I want you to understand. So now the question is, what apart from reproduction is actually a life processes. Digestion is also a life processes. So what make digestion to be a digestion is because of the proteins. That is why we have digestive enzymes. The enzymes that are set, or that the enzymes that are responsible for digesting carbohydrate are different from that of the proteins. So it is very, very important, ladies and gentlemen, that you should understand that proteins are very essential. And as I said from the beginning of the lecture, our life depends on the proteins. So everything that we are doing. In fact, even now that I am talking is because of the proteins that I have. And the more you produce that proteins in your mouth, the more you talk. That is why some people are talkative. And some people they don't speak always. And it's because of the proteins. We see because of our proteins. There are crystalline and opsin proteins. Rhodopsin, they are all found in the eyes, and that is why we use eyes in seeing, and it's because of these proteins. And you will have an eye problem if you are, or if the proteins in the eyes are having issues. So you should get that, ladies and gentlemen. So after this, we have what you call. After this, we have the next thing that we are going to look at is the amino acids. So what do you need to know about the amino acids? So we should know that amino acids, amino acid is a building block of a proteins, as we said, but you should understand that how the amino acid is. The amino acid is made of three components. The amino acid is made of three components. So what are those components? Sorry, four major components. And those four major components, there are carboxylic group attached to it. If you now look at this here, you can see that we have COOH. This is carboxylic group. This is carboxylic group. And then we have amino group where we have NH2. And then we have R group. And then after the R group, then we have free hydrogen. So now if you look at the carbon here, you see that here alpha carbon is clearly written. Alpha carbon is clearly written. So what does that mean? It means that if you look at this carbon, you will realize that in this carbon, there are four different groups attached to it. Look at it, the carbon with that free hydrogen. So that is to tell you, that is to tell you that the amino acids has two major functional groups. The amino acid has two major functional group. And what are those functional group? Is carboxylic functional group and amino as an amino functional group. And one very interesting thing that we should understand is that there is what you call an alpha carbon. So what is an alpha carbon? An alpha carbon is a carbon that have four different groups attached to it. It's a carbon that have four different groups attached to it. So what are the four different groups that attach to this carbon? One is this carboxylic group. If you look at it clearly, we have carboxylic group. This C carbon to oxygen double bond and OH group, so it's carboxylic group. That is, so it's one group. And then we have NH2. We have NH2, which is amino group. And then we have R group. And then we have free hydrogen that attach to it. So this is the alpha carbon. So that is why we have what you call alpha carbon. So this alpha carbon has so many names. In some textbooks, they will tell you stereogenic carbon. In some textbooks, you will see that it is called chiral carbon. So you can either call it stereogenic carbon, alpha carbon. You can also call it chiral carbon or stereogenic center. So all this name is a name given to the carbon that have four different groups attached to it. And then the next thing after this is, so with the exception of glycine, where R is hydrogen, so amino acids have a stereogenic center at the alpha carbon. So it's only glycine, because in the glycine, the R is hydrogen. So if you have R as a hydrogen, it means that this carbon, apart from this free hydrogen, then since the R group is also a hydrogen, so it means that you cannot longer call it, you cannot actually, or you cannot longer call it stereogenic center. You cannot call it stereogenic center. Then the next thing, so all amino acids except glycine, all amino acids except glycine, they have what you call optically active, or they are called optically active. So when we said a molecule is optically active, so then the next thing is all amino acids. So generally, when we said amino acids are optically active, it means that usually they have two types of isomer. They have two types of isomer. And these two isomers, we have what you call L isomer and we have D isomer. So, but which types of isomers most of our amino acids are? They are usually in the form of L configuration. So that is why they are called L isomer. It's also similar to in the case of carbohydrates. The types of carbohydrate that are found, especially in humans, is D-glucose. Is D-glucose. So, but now, what of in the case of amino acid? It is L. It is called L isomer. So it's L isomer that are found in humans, but some microorganisms have D isomer of the amino acid. But most of our amino acid, especially in humans, we have L isomer. L isomer of the protein. So this is very, very important. And then note that the Fisher convention used with carbohydrate is also applied to amino acids. So we have what you call Fisher projection, and this is a Fisher projection. This is how to write amino acids in a Fisher projection form. So you see now, this is actually an L glyceraldehyde, that is in the case of carbohydrate. So our amino acid is also in the form of L amino acid. So why it is called L? It means that L means laboratory. L means laboratory, while D means dextrorotatory. D means dextrorotatory and L means laboratory. So therefore, now if you look at this amino acid, the one that is in L isomer, look at the amino group. The amino group here, is it at the left or at the right? It is at the left. So usually when the amino group is at the left, usually if the amino acid is at the left, then it is called L isomer. And if the amino acid is at the right side, it means that it is called D isomer. So if the amino acid here is at the left, so it's called L isomer. And if it is at the right side, it's called D isomer. So you should understand that as I said, we have L isomer and we have D isomer. So L isomers is the form of the amino acid that are usually found in humans. So any amino acid that doesn't have L isomers, like for example, if we take anything that have L isomer, or if we take anything that have D amino acid, even if we take it, we cannot digest it. But how do we digest it? Because we don't have the enzyme that digests D amino acid or that process D amino acids. But we have some intestinal microorganisms that have an enzymes that can do that. So, and most of those intestinal microorganisms, they're extracellular organisms, so they usually excrete the enzymes in our GIT, and then the enzymes will now help us in processing the amino acids in the D form. But we don't have that enzymes, unless using our intestinal microorganism. Okay, so then now as we are saying, so look at it. This is how the amino acids look like. So we have it like this. The one is at the left and the right hand. So now the amino acids are known by a common names. So each also has a 3-letter abbreviation. So each amino acid has a 3 letters abbreviation and also a 1-letter abbreviation, which used to describe the amino acid sequence in a protein. Because as we said, the proteins is made up of an amino acid. So now the question now is, how do we represent the amino acid in a linear chain, in a linear change of a protein, or in a protein? How do we represent them? You cannot start by writing the full name of the amino acid. It will be very difficult. It will be very, very difficult to do that. So that is why there are 2 ways that you can shorten the name of the amino acid. You can either use 3 letters abbreviation or 1-letter abbreviation, so that you can actually write the sequence of the proteins. Okay, and then the next thing after this is so this is how the amino acids look like. We have glycine, we have alanine, we have valine, we have, so these are the kind of the amino acid, and they have their letter of abbreviation. Like for example, glycine has a 3 letters. So the 3 letters of the glycine you see is GLY, is GLY. And of course, the single letter is represented with capital G. And then alanine is ALE, and then the single letter is A. Valine is VAL, and the single letter is Y. Leucine is LE, and the single letter is L. So now, if you check here, you can see clearly is one amino acids or one amino group and one carboxyl group. So these are the amino acids that have only one amino acid and one carboxyl group. So they are the one that have this. So we have serine SER represented with capital letter S, and then we have threonine with capital letter T. So all these amino acids, this is their formula. This is how to draw their structure. Some of you are complaining that the write-off is very tiny. Is that applied to everyone? I think the problem is maybe from your phone. I'm asking. You can't see it clear. So what do? You are saying that the write-off is tiny. Okay. So, but can you now, are you now seeing it from here? So I think let's just try to manage, and I think after the class, maybe I will send you slides that will address that, because the most important thing that you should understand from from the this tiny slides that you complained is that uh, the most important thing is to understand that the amino acids can be represented in two ways. You can use a three letters and also a single letter. So that's the most important thing for this slides. You can use a single letters, or you can use a single letter, or you can use a double letter. Sorry, a triple letters. Like for example, glycine. Glycine can be it's written uh, is GLY, and the single letter is G. So after this class, I'm going to send you maybe an image that will address that. So that is the most important thing that you should know there. So then now the next thing is the classification of amino acids. So amino acids are classified on different bases. We have essential amino acid, we have of course non-essential amino acids. So what are the essential amino acids? So 8 amino acids cannot be synthesized by adult human and therefore must be included in the diets. So From this slide, the most important thing that you should understand is that uh, under the classification of amino acid are classified into essential and non-essential. So when we said essential amino acid, it means that there are those amino acids that we cannot synthesize them. As an adult, we cannot synthesize the amino acids. So it means that we have to get them from the diet. And we have non-essential amino acids. So non-essential amino acids are the amino acids that our body can synthesize them. So our body can synthesize this type of amino acids. And then we have them here. So we have 8 amino acids that are essential, and we have 12 amino acids that are non-essential. So the most important thing that you should understand at this point is that generally we have over 300 amino acids. We have over 300 amino acids. But the most important ones that are found in proteins, the most important ones that are found in proteins are 20. We have 20 of them that are found in proteins. So now, it's out of these 20 that they are classified into two: essential and non-essential. So when we said non-essential amino acids, we have 12 and essential are 8. So we have valine, leucine, isoleucine, threonine, methionine, phenylalanine, and tryptophan, and then finally lysine for the essential. While for the non-essential, we have glycine, we have alanine, serine, proline, tyrosine, aspartic acid, glutamic acid, asparagine, histidine, and aspartate. They are example of non-essential amino acid. That is the one that we can synthesize them in our body. So this is the classification of amino acid based on their destiny. So we have what we call acid-base properties of an acid. Acid-base properties of an amino acid. So generally, if we look at the structure, if we look at the structure of amino acids, it has both carboxylic group and amine functional group. It means that it has both acidic functional group and it also have basic functional group. So the carboxylic acid and amine functional groups are simultaneously present in amino acid, and we might ask whether they are mutually compatible since one group is acidic and the other one is basic. So amino acids have both basic functional group and acidic functional group. But now the question is, are they compatible? So generally, amino acid with one amino group and one carboxylic group are better represented as a dipolar ions. So the best way to represent these amino acids is to write them as a dipolar ion. So when we say dipolar ions, it means that remember that an acid is a substances that donate a protons, while a base is a substances that accept a protons. So you see now clearly, if you can see in the ionic form, this is the amino acid. So you see in this case, you have carbon to oxygen double one, and you have free oxygen here with negative charge. So that means that the oxygen here, remember from the beginning, this oxygen with negative sign, it has hydrogen. So because it is an acid, so it means that it donates a proton. And then of course the NH3, you are still complaining that it's not clear. So if you now check here is NH3. So in the NH3, remember initially it is NH2. So now it is NH3 because the NH2 accept the proton. So that is why it is called a base. It is called a base. because it is now NH2, so it accepts a protons, while the COOH donated, it donated, it donated a proton. So that is why it is called an acid. The COOH is acid, and then NH2 is the basic because it accepts a proton. So generally, amino acid A and P are amphoteric. Amino acid are amphoteric. When we said amino acid are amphoteric, what does that mean? It means that they can behave both as acidic because they donate a protons to a strong base, and they can also behave as a base because they accept a proton from the strong base. So that is why you should understand that an amino acid is an is amphoteric because it has both acidic properties and basic properties. So how does it have acidic properties? When it donates a protons. And then when it accepts a protons, then it has a basic properties. So we have what you call isoelectric point. So isoelectric point, that is Pi, is when the amino acid will be dipolar and have a net charge of 0. So when we said isoelectric point, it means that it's a point where the net charge of the amino acids is 0 and it is dipolar. So what does that mean? If the COO is negative and the NH2 is positive, it means that the negative sign from the COO and the positive sign from the NH3, they will neutralize it. They will neutralize it. So if they neutralize it, it means that the net charge is going to be 0 because plus minus, it is going to be 0. So an isoelectric point is a point where the net charge of an amino acid is 0. So that is what you call acid-base properties of an amino acid. Acid-base properties of an amino acid. And then the next thing now is the synthesis of amino acids. Please, since you are having problem with some of the slides, I am now going to end this, and then I will resend another link so that we will join another lecture so that we are going to show more clearer slides. Or if you think maybe you can stop the class from here, then we will continue maybe on Saturday. But what is your suggestion? Can we continue with the class? Maybe we change another slides. I am asking. Can we rejoin with another link? Okay. So let me just get another this. So I'm going to send you another link. I'm going to send you another link. Okay. I'm going to send another link.